Assalamu alaikum. Good morning. I am Dr. Esraa from the Gastroenterology Department, and today I'm trying to take a brief talk about the mitochondrial dysfunction in the inflammatory bowel disease. So, as it's all known that the inflammatory bowel disease is a chronic case, and it's complex with multisystemic fiction, and it's relapsing and remitting course. The inflammatory bowel disease pattern is often like a cyclic pattern. Sometimes the patient come with active inflammation and sometimes in remission cases. And the severity also differs. The main two inflammatory bowel diseases is the Crohn's disease and the ulcerative colitis. And both of them differ in the involvement of the GIT. The Crohn's disease, it can affect any part of the GIT, even the upper gastrointestinal tract, like the stomach and the esophagus or the duodenum. But the ulcerative colitis only in the lower part, in the colon and the rectum. The other difference is the Crohn's disease affects the transmural inflammation, all the layers of the intestine, but the ulcerative colitis, it's only mainly in the mucosa. The severity also depends upon the location and how severe the inflammation is involving the disease. In these pictures, we can see during the endoscopy how we can diagnose the Crohn's disease. It's appearing like patchy areas with aphthoid ulcers or aphthoid erosions, like here. Sorry. How can we come back? I need to go back. Okay. Here we can see the aphthoid ulcers, and here also, and here it's in the ileum. So this is different part of the colon. As in this figure, we can see that it can affect many different parts in a patchy area, not a continuous manner. And in this slide, we can see when it affects the duodenum. Other extraintestinal manifestations we can see, like the patient complaining of symptoms like aphthoid ulcers in the gingiva, and also we can see in the ulcerative colitis, it's not like a patchy area distributed inside the colon, but we can see it all over the colon itself. And also this is cobblestone appearance. It happens with the lower part of the colon and the rectum, and even some patients coming with inflammation in the anus area, so we can see proctitis and also inflammation outside in the external anus sphincter. This is the main two difference between the ulcerative colitis and the Crohn's. The ulcerative colitis also not affecting the upper intestinal tract. The other symptoms we can see, like regarding the GIT, the patient complaining of diarrhea, abdominal pain, loss of weight, and some blood or mucus in stool. The extraintestinal manifestations we can see mainly in the Crohn's disease, like the joint pain, also mainly in the lumboiliac spine and the mouth sores, sometimes coming also with red eye symptoms, which calls it uveitis. So the pathophysiology of this disease, it's multifactorial. It's including the genetics, the environmental factor, and the immunoregulatory factors. Today we will discuss about the role of the mitochondria, how it could affect the inflammatory bowel disease. To know the mitochondria was firstly discovered by the pathologist Dr. Richard Altman in 1890, and the name was coming from threads and granules disease. It's a Greek name, mitos like threads, and chondrion is a granular like shape, and it's a double layer rod shape, and we can see like outer membrane, inner membrane. The inner membrane with folds like it's crusty, and the intermembranous space. The outer membrane and the inner membrane mainly contain the phospholipids and proteins, and the inside the inner membrane, many chemical reactions and processes take place, and the production of the ATP molecules, which is the main function of the mitochondria. And inside the matrix, we can see the different parts involved in the processes taking part in the mitochondria, like the granules, the DNA, and the ribosomes. The mitochondrial DNA, there is an important information about it, that it's only inherited from the mothers, because during the fertilization of the oocytes, we can take our mitochondria inside the nucleus. It's kept in the oocytes, but from the sperm, it's in the neck, which is not involved in the fertilization process. So the mitochondrial DNA itself, it's only inherited from the mother sites, and different than the nuclear DNA. So every one of us has two DNA: the DNA in the nucleus, which is the main DNA, and other DNA inside the mitochondria. The mitochondria is it is circular in shape, and if every mitochondria have many copies inside it. Talking about the mitochondrial genome, it's encoding about 13 proteins and 22 transfer RNA, 2 ribosomal RNA. All of this needed to translate these proteins within the mitochondrial matrix to be essential for the oxidative phosphorylation system, which is the main process for producing the ATP. The other DNA inside the nucleus, encoding many proteins, and also affecting the mitochondrial dynamics. It's synthesized in the cytoplasm, and after that transferred inside the mitochondria. So we can conclude that the mitochondria is controlled by two DNA: its DNA itself and the DNA in the nucleus. So how can we inherit this mitochondrial DNA? It doesn't follow the regular Mendeleyev trace like the autosomal recessive or dominant or X-linked pattern. Otherwise, it's follow the mitochondrial inheritance from the maternal line. Complex trait means multiple genes interacts with each other and interacts with the environmental factors. Sometimes it happens like a de novo mutation, means that it's a new mutations happening in that person for the first time, not detected in each parents or in one of the relatives. In our inflammatory bowel disease, if there is any mutation happens in the mitochondria, all in the nuclear DNA, so it will follow this three parts, and mainly the complex part. It will not follow the Mendelian traits. So far, the genome-wide association studies, this is the test used to detect like the whole genome of the person, detected more than 160 genetics could be susceptible for the inflammatory bowel disease. The exact nature of this genetic predisposition is still complex, because it interacts with other several genes. Some several genes could considered like a protective genes against the disease. Some genes considered like flaring up the disease, and also not interacts with each other alone. It interacts with other factors like the environmental factors and the immunology system. Like we can see in this figure, there is some examples of the genes could be having mutation in it. If the patient have one of these genes, it could interact with any bad habits in the environment around him, like bad diet, he's heavy smokers, taking frequently antibiotics, or heavy NSAIDs consumption. With this environmental factors, he could impair his immunological system, and this immunological system will impair the epithelial barrier cells in the intestine, and it will attack the epithelial cells, causing the disease. And it also will alter the regular microbiomes inside the intestinal lumen. For example, to prove our theory that the genetics not only the main role in this disease, if we have two identical twins, so two identical twins, they are sharing the same genes, but they are exposed to different other factors. Some exposed to these factors like smoking, NSAIDs, antibiotics, or infections, or any other factors, and some not exposed. So this individual exposed to these factors will have increased risks for developing the disease. The other group, it will have lower risk to develop this disease. So the role of our mitochondria, it's the main powerhouse of the cell. So it's the main role of the mitochondria is to produce the ATP. And this ATP, this is the energy used, processed by different cells in our body to affect his functions, and mainly during the oxidative phosphorylation process. This is not the only role. They are integrated in many metabolism pathways in these cells, but this is the main role. And the other processes they are involved in, like promoting new cells and new cells formations, multiplications, even the apoptosis, metabolic signals, thermogenesis of the body, regulates other metabolic activities like cellular differentiation. And this is necessary that mitochondrial dynamics and the biogenesis, the function and the structure, is controlled, should be tightly controlled to maintain the overall cellular homeostasis in the normal individuals. In the following slides, I will discuss main 6 processes should be tightly controlled in the mitochondrial function and in the intestinal lumen and function to have a normal individual without inflammatory bowel disease. Any alteration of this following processes, it will cause the disease. So we'll understand how the pathophysiology of the affected mitochondria could cause the inflammatory bowel disease. The first thing is the tight junctions between the epithelial cells itself and the intestinal barrier between the lumen and its content and the epithelial cells. If there is any alteration in the tight seals between these junctions, could affect the barrier cellular space and could affect the gut barrier, become more permeability, and the barrier will be incompetent to prevent the antigens and the harm organism to get inside the gut and get inside the epithelial cells. The second thing is the beta oxidation process. This beta oxidation process, it's used to degradation of the butyrate. The butyrate is a natural nutrient found in the food and produced by intestinal fermentation of the dietary fibers by the gut bacteria. And it's important to providing over than 70% of the energy needed by the colonic epithelium. If this beta oxidation process decreased or affected because of the dysfunction of the mitochondria, it will affect the energy produced to the epithelial to be normal functional. The third process is the intestinal epithelial itself could be compromised, it's not renewed as much as is needed with the turnover. Every 4 to 5 days in the normal individuals, it's necessitating to be renewing the damaged epithelial cells during the normal digestion processes. And this needs huge supply of energy. If there is any defect in the mitochondria, so there will not enough energy to renew this damaged epithelial cells, and it will affect the absorptive enterocytes. It will affect the mucus-producing goblet cells and the cells for the endocrine functions, and the Bannet cells was also important for the secreting functions. A study was found, a mutation in NOT2 gene, it could affect and cause Crohn's disease by affecting this process. The fourth thing is the disruption in a process called mitophagy and autophagy. Mitophagy is the autophagic response which is targeted the damaged and cytotoxic mitochondria, because mitochondria during the controlling the normal function in the body, it cause be damaged, and this has to be regularly removed. It's removed by the mitophagy process. If there is any defective in the mitophagy process, it will accumulate the damaged mitochondria, and it will accumulate also reactive oxygen species, which is causing oxidation of the cells and damage. The GWAS, the genome-wide association test, it detected three genes could be related to defect in this process, and also related to the Crohn's disease. The fourth process is the mitochondrial communications, means the mitochondria could communicate with the nuclear DNA outside with a mitochondrial nuclear crosstalk. It's called like mitochondria unfolded protein response, because as we said before, that the mitochondria is controlled by its DNA and the outside DNA. If this pathway disrupted by a gene mutation, it will be affect the function of the normal individual intestinal lumen, because it's very important in re-establishing the protein homeostasis in the process. The final thing is the reactive oxygen species and the nitrogen species. If these two products, it coming out from the different metabolizing processes inside the cell. If there is any accumulation in these two without proper turnover from them, it could causing oxidation of the proteins, lipids, DNA, and alter the biological function and cause cell death. This will explain more what we said now. This is a picture of the normal intestinal. So the outside things here, there is the bacteria, and this is the human antigens. It's protected against the epithelial cells with this yellow shaded mucus layer. The mucus layer by the goblet cells, so to protect the intestinal epithelial cells. The second important thing is the Baneth cells. Baneth cells is secreting epithelial cells, and it releases this blue stars organelles. This is antimicrobial peptides. And the mitochondria. Here the mitochondria, it's look healthy in structure with its crusty, and this tight junctions, it's normal without any impermeability in it, so it's prevent any permeability of the harmful antigen and the bacteria inside the cells. The cellular antioxidants, this is the blue circles. It protects the intestinal epithelial cells with the reaction with the oxygen species. So they coat the reactive oxygen species to prevent the cells from the oxidation effects. And this is the leukocytes is doing survey for the lamina propria from any harmful organism could be permeable inside. Now it started to be diseased cells. The diseased cells started with decreased the antimicrobial peptides, the blue stars, and increased the mucus layer. This exposed the cells from entering the microbiota and the antigen to be inside the cells and start to be harmful and damaging. And here also we can see that when increased the epithelial permeability, so because of the decreased cellular antioxidants, the antioxidants here is now much less compared to the normal one. So and we can see that the reactive oxygen species become more numerous because it's not turned over by the antioxidants, and the inflammation, the infiltration of the immune cells, because the body start to react to this infiltration of the harmful organism. So the immune system start to react by many immune cells. It will start not only damaging the antigen, it will also damaging the epithelial cells. the mitochondria here, it's having genetic defects and mutations, so it's not healthy and reducing the ATP production and increase also the reactive oxygen species, which cause oxidations. The main immune cells, the main immune mediators released is the interleukin 8 and interleukin 1b, and which also flare up more pro-inflammatory mediators. So we can now conclude that when all these main processes defects, the damage will happen to the epithelial cells, and it will happen its normal functioning, and it will become ulcerated. And also the immune system, when it's activated, it will not only damaging the epithelial cells, it will also damaging the multi-systems we can see in the patient who come is symptomatic. So the inflammatory bowel disease, so far, it's how could be managed. We can diagnose it with the EGD colonoscopy, even capsule endoscopy, if we can see if we couldn't see anything during the colonoscopy. With the imaging, we can see in the CT or MRE angiography if there is any thickening or there is any fistula. By the stool analysis to see inflammatory markers like the calprotectin and the occult blood. In the blood, we can see like the serology for ANCA, which is inflammatory reactions. We can see it increased in the patient affected. For the blood investigations, other blood investigations to detect any other complications like the anemia, the main thing, because of the loss of the blood, because of the chronic inflammations. And the treatment mainly with aminosalicylates, with steroids during the flare-up and during the relapse, immunomodulators in severe cases, and the biological treatments. Surgical treatment is mainly in the patients with severe complications, perforation, strictures, fistula, or resection even of the diseased part, not responding to the treatment. To summarize our topic today, because it's like very basic information that could be a little bit complex, that our inflammatory bowel disease, it's not a single factor caused it. It's a genetics, mainly following the complex trait pattern, environmental and immunoregulatory factors. And a variety of conditions and stimuli could alter the mitochondrial structure and function, and this will affect the overall cellular function and tissue organ function. The mitochondrial structure and function is controlled by a cellular DNA and the mitochondrial DNA. However, the genetic predisposition alone is not the only factor for the inflammatory bowel disease, but it has an important role. And we still need more studies to understand the exact pathophysiology mechanism and the pharmacogenetics response for the treatment, so we can precise more management plans suitable for each individuals in the future. Thank you. Very, very